So here's what happens. If a cell gets infected by a virus, and the viral antigens are expressed on the surface, it can be killed. And NK cells can also help with macrophage activation because they can make interferon gamma and activate macrophages to some degree. But that's not their major role. Their major role is killing the virus-infected cells. These are called large granular lymphocytes because they're larger than T or B cells, and they've got lots more granules for killing. But they use the same killing mechanisms as cytotoxic T cells. They make perforin, they have granule enzymes that can leak through the perforin pores, and they secrete lots of tumor necrosis factor alpha, which will kill the cells as well. So they use the exact same toxic mechanisms as a cytotoxic T cell does, but they've got no antigen receptors. They can't recognize a virus antigen from a non-viral antigen. So how do they know what to kill? Well, that's illustrated here. This is this is a little bit more detailed picture than what you have in your notes, but what you have in your notes is quite sufficient. Here's the story. NK cells have two different types of receptors, an activating receptor and an inhibitory receptor. There are at least 6 or 7 different activating receptors. A couple of them are named there. You do not need to know the names of these things. They have very bizarre acronyms, and you're not expected to know those. If you wish to memorize them, that's fine with me, but you'll never ever see them again the rest of your life. So it's up to you. You can memorize it or not. But if I were you, I wouldn't. These activating receptors recognize common glycoproteins expressed on all nucleated cells. At least because there are several of them, at least one is on every nucleated cell. So that provides a positive signal. That's what that little plus sign is. It provides a positive signal to the natural killer cell and says to the natural killer cell, hey, you just bound a nucleated cell. Only nucleated cells express these molecules. You just bound a nucleated cell. You could kill it. And the NK cell says, oh good, I get to kill something. But then, if the cell has a MHC class 1 molecule on it, HLA or HLA-B or HLA-C, and so forth, as you see, it interacts with what is called a killer inhibitory receptor, KIR. And there are a couple different ones of these, but there's one major one called the killer inhibitory receptor. And that sends an overriding negative signal inside of the NK cell saying, wait a minute, this one's got MHC class 1 on it, you can't kill it. So the NK cell goes, I can't? Darn. Oh, hey, here's another nucleated cell. I'm going to kill this one. It's got class 1 on it too. Here's another one. It's got class 1 on it too. They lead a very frustrated life because every time they come in contact with a cell, they get one signal that says kill it, and then they get another signal that says, hey, abort that. You can't kill it, it's got class 1 on it. So, but every now and again, in the second panel, the NK cell gets lucky. It comes in contact with a virus-infected cell that was infected by a virus that turns off MHC class 1 expression, like we saw here. Or, no, wait a minute, here, yeah. Like, say, one of those herpes viruses. So it gets the activating signal, because the activating glycoproteins are there, and it says, okay, where's the negative signal? And there's no class 1 there. It says, oh, there's no class 1 in this cell. It's a nucleated cell. It's got no class 1. It kills it. Just kills it. The virus can't win. If the virus allows MHC class 1 expression, the viral peptides are bound to class 1, and the cytotoxic T cell kills it. If the virus blocks MHC class 1 expression, the natural killer cell sees the cell, says, hey, there's no class 1 here, and then it kills it. Now, natural killer cells ignore red blood cells. Red blood cells have no nucleus. They don't have class 1 molecules on them. They're the only cell in the body that lacks MHC class 1 under normal circumstances. But they also don't have the activating molecules either. So it'll ignore normal red cells. But any other nucleated cell, it won't kill unless the class 1 molecule has been turned off by a virus infection trying to escape cytotoxic T cells, and then the NK cell kills it. Pretty cool. Here, this is all shown in one slide here. I forgot I had this slide. Now, we have a third killing mechanism that's available called antibody-dependent cellular cytotoxicity, or antibody-dependent cell-mediated cytotoxicity, ADCC. This happens when you get an infected cell which where the infecting agent, like say a virus, produces viral proteins on the cell surface. Now these aren't little peptides bound to MHC class 1 molecules or class 2 molecules. These are the intact viral antigen. Many viruses have to put viral glycoproteins on the cell surface in order to replicate when they bud out of the cell and take some cell membrane along with them. So this is a quite common occurrence in viral infection. But of course that's a foreign antigen, which is extracellular, it's on the outside. So you can make antibody against it. Now the only antibody here that works is IgG. That's the only antibody that mediates antibody-dependent cellular cytotoxicity. And if you look back on my antibody lecture, you'll see it's listed that way. So what happens is that NK cells, which have Fc receptors, which recognize the Fc end of IgG, will bind to the IgG, the purple molecule that's bound to the viral antigen on the cell. That triggers them to kill the cell. That's antibody-dependent. In fact, the CD16 molecule, which uses a marker for NK cells, is the Fc receptor. That's a CD molecule that identifies NK cells. There's another one called CD56. Both of those together are used to identify NK cells. And they are two CD molecules you need to know. NK cells are pretty important cells. So they will bind to the Fc end of IgG. That will trigger them to kill the cell. And this one is independent of MHC class 1. This cell can have a ton of class 1 molecules on it. That would not inhibit this killing. You can inhibit antibody-dependent cellular cytotoxicity by anything. If there's IgG bound to the cell, the NK cell will kill it. Now, other cells mediate ADCC as well. NK cells can do this. Macrophages, eosinophils, even neutrophils can do this, but they're very inefficient because they don't make perforin and granzymes that the NK cell does. So it's got the most powerful killing molecules for killing a nucleated cell. It's designed to do that. So it's the most efficient cell at mediating ADCC. So the picture in the bottom really kind of illustrates it pretty nicely. An IgG antibody bound to a that's for phagocytosis, but the top picture really illustrates the ADCC pretty nicely. So in summary, we have activation of macrophages, which kills pathogens and inside the macrophage. We have cytotoxicity, which kills pathogens that try to replicate in the cytoplasm. And we have ADCC, which kills cells which express antigens on the surface that you can bind antibody to. And then we have natural killer cell killing. Now, why wouldn't cytotoxic T cell killing and NK killing be enough by themselves? Well, there's actually one virus that's figured out a way around both killing mechanisms. This is not in the notes, but I got to tell you the story because it's just too good a story to pass up. Cytomegalovirus, which is one of the herpes viruses. CMV escapes cytotoxic T cell killing because it very efficiently turns off MAC class 1 synthesis. It's got 3 separate genes that block MAC class 1 synthesis, MAC class 1 transport through the cell, and it accelerates MAC class 1 digestion within the cell. So it really doesn't want MAC class 1 expressed because it replicates fairly slowly. And the T cells would wipe it out if they got anywhere near its infected cells. So it shuts off class 1 synthesis, escapes cytotoxic T cell killing. But now it's got to deal with the NK cell, and it escapes the NK cell killing too. How does it do that? It has another gene that puts a fake MAC class 1 molecule on the surface, a decoy molecule that looks a little bit like class 1, but is really a viral protein. And it puts that on the surface of the infected cell instead of the real class 1 molecule. And it works. It fools the NK cell. The NK cell gets a negative signal from this molecule and doesn't kill the cell. But we've got ADCC. That fake molecule is a whole intact viral antigen. Well, you can make IgG antibody against that, which is what you do. The IgG binds to the decoy molecule or other virally expressed surface molecules, and ADCC kills the virus, infected cell. So even if it escapes the first 2 mechanisms, ADCC gets it. The virus can't win. We